Hello everyone, welcome to Biology Tips. In today's session, we will discuss about transcription. Transcription is a process which is required in plus 2 level as well as in degree level. So, what do you mean by transcription? The process of copying genetic information from one strand of DNA to RNA is called transcription. This is the easiest definition. What is transcription? The process of copying genetic information from one strand of DNA to RNA. So it is a process by which the information which is present in DNA, it is copied to RNA. And we have to make three types of RNA: tRNA, mRNA, and rRNA. When you look at DNA, we have two strands. One is running in 5' to 3' direction, and second one is running in 3' to 5' direction, right? Now, when you create RNA, we will be able to create only one RNA from a DNA. Okay, so the 5' to 3' strand, we will call it as the sense strand or the coding strand. 3' to 5' strand is called as the antisense strand or non-coding strand. Now, as it is given in this picture, we will be always creating any molecule in biology in 5' to 3' direction. So this will be synthesized opposite to 3' to 5' direction. Okay, so that means opposite to antisense or non-coding strand will be synthesizing RNA. Okay, so when you look at transcription, what is transcription? It is the process of copying genetic information from one strand of DNA to RNA. We have two strands present in a DNA: non-coding strand or antisense strand and coding strand or sense strand. Non-coding strand or antisense strand is running in 3' to 5' direction. This is the one who will serve as a template and produces the working copies of RNA molecule. The coding strand or sense strand, which does not participate in transcription. It is running in 5' to 3' direction. Now look at this picture. Here you can see that I have given 5' to 3'. This is a coding strand. 3' to 5', this is the non-coding strand. Opposite to this, we have created the mRNA. Just look at the codons. You can see the codons on mRNA. Opposite to T, A is present. Opposite to A, U is present. Opposite to C, G is present, right? Now, when you look at the coding strand, the sequences ATGCA, CGCAC, it is same as that of mRNA. The difference, in place of T, U is present, right? So what is the speciality of coding strand? With the exception of thymidylate for uridylate, primary mRNA contains codons with the same base sequence. Okay, so this part is important for entrance examination. This is important for your board examination also. When you look at transcription, transcription is the process of copying information which is present on a DNA to RNA. Now, we have two types of strand in a DNA: coding strand and non-coding strand. When you look at coding strand, the sequences which is present on DNA is same as that of RNA, except in place of T, in RNA, we will have U. When you look at non-coding strand or the antisense strand, right? This is acting as a template on which we are creating the mRNA. Now, the sequences which is present in mRNA will be complementary to that of which is present in non-coding strand. Okay. We have to study about a transcription unit. What do you mean by a transcription unit? The segment of DNA that takes part in transcription is called transcription unit. It consists of three regions: promoter, structural gene, and terminator. So what do you mean by transcription unit? The segment of DNA that take part in transcription is called transcription unit. It consists of three regions: a promoter, structural gene, and the terminator, right? Now, you know what is template strand. Template strand is running in 3' to 5' direction. Opposed to this, the mRNA will be formed 5' to 3' direction strand, we will call it as coding strand, right? Now, in depth, we will study about transcription unit. First one, promoter. Promoter, can you see? When you look at the template strand, this is present 3'. Whereas when you look at coding strand, this is present on the 5' end of coding strand. Promoter is the region which possesses some recognition sequence. This is a region where RNA polymerase will come and bind. So what is promoter? It is located upstream of structural gene at the 5' end of coding strand and 3' end of template strand, right? 5' end of coding strand, 3' end of template strand. It is a site for binding of RNA polymerase. It is the presence of a promoter in a transcription unit that defines a template strand and coding strand. Okay? So promoter, structural gene, terminator. All these points you should know, then only you can write this essay. Now, what is terminator? Terminator is present downstream, right? It is present in the 3' end of coding strand and 5' end of template strand or the non-coding strand. It usually defines the end process of transcription. Once the RNA polymerase starts its activity, once it reaches terminator, it will stop transcription. So what is terminator? It is located at the downstream of structural gene, right? After structural gene, it is present on the 3' end of coding strand and on the 5' end of template strand. It usually defines the end of transcription process. Moving on to the next session, we are going to study about structural genes. So what is structural gene? It is a component of DNA strand which codes for information. That means this is a region which carries information. From this, mRNA will be produced. Now, once the mRNA undergoes translation, it will be producing the proteins. Okay. So, a transcription unit is a region of DNA which undergoes transcription. It possesses 3 parts: promoter, structural gene, and terminator. Promoter is the region which is present on the 3' end of the template strand and 5' end of coding strand. Right? Now, promoter is the region where RNA polymerase will come and bind. Terminator is the region which is present on the 5' end of template strand. After structural gene it is present and 3' end of coding strand. This is the region which defines the end process of transcription. Now, structural gene, this is the region which will code for information. From this region, we have to produce the RNA, which carry information. When you look at transcription, we cannot avoid RNA polymerase, right? RNA polymerase is the enzyme which helps in transcription. Polymerase, as the name suggests, it helps in polymerization reaction. Now, why it is called as RNA polymerase? Because it produces RNA. Right, nucleotides join together to form a nucleic acid. So it's a polymer. Now, this RNA polymerase we will call it as DNA-dependent RNA polymerase. Why? Because 3' to 5' end of DNA, which is acting as the template strand. On this template strand, we are creating another RNA, right? So its activity depends on a DNA strand. So we are calling it as DNA-dependent RNA polymerase. RNA polymerase, RNAP, or ribonucleic acid polymerase is a multi-subunit enzyme that catalyzes the process of transcription where an RNA polymer is synthesized from a DNA template. So look at this diagram. Here you can see that the RNA polymerase enzyme, it's having many subunits, right? 2 alpha subunits are there, 1 beta unit, 1 beta prime unit, then there is an omega unit. This part we will call it as the core enzyme. So the statement is true. It's a multi-subunit enzyme, right? Now what is its function? Production of RNA primers. The sequence of the RNA polymer is complementary to that of the template DNA and is synthesized in 5' to 3' direction. Okay. I hope you are able to follow. RNA polymerase structure. When you look at the structure of RNA polymerase, it's a multi-subunit enzyme. Now, a single RNA polymerase transcribes all type of RNA in prokaryotes. RNA polymerase core enzyme consists of 5 subunits, 2 alpha, a beta and a beta prime subunit and a small omega subunit. Okay. A sigma factor binds to the core enzyme, forming the holo enzyme. So you can see when the sigma factor will come and bind to this core enzyme, it will form, right, the holo enzyme. So this is the core enzyme. The sigma factor binds to this core enzyme, it becomes the holo enzyme. A sigma factor binds to the core enzyme, forming the holo enzyme. After transcription starts, the factor can unbind and let the core enzyme proceed with its work. The core RNA polymerase complex forms a crab claw or clam jaw structure with an internal channel running along the full length. So once this RNA polymerase core enzyme is there, it can act as a crab claw, right? There will be a channel in the center. Through that, the nucleic acid will be moving. Okay, so this is about the structure of RNA polymerase. I hope you are able to follow. Moving on to the next session, we are going to study about RNA polymerase types. We have 3 types of RNA polymerase enzyme: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 transcribes 28S rRNA, 18S rRNA, and 5' rRNA. RNA polymerase 2 transcribes mRNA. RNA polymerase 3 transcribes tRNA, 5S rRNA, and snRNA, small nuclear RNA. Okay, this part is highly important for the entrance examinations. So how to study this? Look at RNA polymerase 2, right? TWO2. Now when you take ulta of W, you will get M. So easily you can remember RNA polymerase 2 transcribes mRNA. RNA polymerase 3 is having T, right? So it directly connected with tRNA. Now RNA polymerase 1, that is the remaining one, it's mainly associated with rRNA, right? So when you look at, we have mainly 3 types of RNA: rRNA, tRNA, mRNA. RNA polymerase 1 is associated with rRNA, RNA polymerase 2 is associated with mRNA, RNA polymerase 3 is associated with tRNA. Okay. RNA polymerase 1 transcribes 28S RNA, RNA polymerase 2 transcribes mRNA, RNA polymerase 3 transcribes tRNA. Okay. Moving on to the next session, we are going to study in depth about transcription. Transcription involves 3 stages: initiation, elongation, and termination. So first we have to study about initiation, right? Now, when we studied about the transcription unit, we studied that in transcription unit, we have 3 regions: promoter, structural gene, and terminator. So initiation is associated with promoter region. In promoter region, we possess 2 sequences. In the case of prokaryotes, which can be recognized by the RNA polymerase. Okay. So here we are using RNA polymerase 2 because we are going to produce mRNA. RNA polymerase bind to the promoter and initiate transcription. Recognition is done by sigma factor, which is also called as initiation factor. There are 2 base sequences on the coding DNA strand which sigma factor of RNA polymerase can recognize for initiation in prokaryotes. One we will call it as the Primno box or Tata box. The sequence is TAT-AAT. The second one we will call it as the -35 sequence. Otherwise we will call it as the TT-GA-CA sequence. Okay. Now look at this picture. Here you can see this is the 5' to 3' end. And this is the 3' to 5' end. The promoter is present towards the 5' end of the coding strand and towards the 3' end of the non-coding strand, right? The first sequence near to the promoter we will call it as -10 sequence, that is Tata box. The sequence is TAT AAT. The second sequence, it is in -35 position, we will call it as -35 sequence, which is TTGAC. Any of the sequence can be recognized by the RNA polymerase. Now the DNA dependent RNA polymerase will come and bind here. That's the way initiation occurs. When you look at eukaryotes, there is a slight difference in the recognition sequence. In the case of eukaryotes, we will call it as the Hogness box and cat box. There are two base sequences in the coding DNA strand, which RNA polymerase can recognize for initiation in eukaryotes. One is Hogness box, otherwise it is called as Tata box. The sequence is TATAAA. This is similar to that of Pribno box in prokaryotes. It is located on the left about 25 nucleotides away from the starting point of mRNA synthesis. The second sequence we will call it as cat box. CAAT. Cat box we will call it as GGCCAATCT. This is the sequence. Can you see here? CAAT is coming here. So it is called as the cat box. It is located between 70 and 80 nucleotides upstream from the start of transcription. So when you look at initiation, it is a step associated with promoter. The DNA dependent RNA polymerase will come and bind to the promoter. Now to bind with the promoter, we require the recognition sequence, which is different in the case of prokaryotes and eukaryotes. When you look at prokaryotes, we have the Pribno box or the Tata box. It is in -10 position. Next one, it is the -35 sequence, TTGACA. When you look at eukaryotes, one sequence which is 25 nucleotides away upstream on the DNA strand, that we will call it as Tata box, TATAAA. Otherwise it is called as the Hogness box. The second one, that is called as the cat box, CAAT box. The sequence is GGCCAATCT, which is located between 70 and 80 nucleotides upstream from the start of transcription. Okay, so these are the important points which you have to remember related to initiation. When you look at transcription, certain keywords are there which you have to remember. Okay. Moving on to the next session, we are going to study about elongation, the second step of transcription. Now, in elongation step, we will be synthesizing the RNA molecule. Okay, so as RNA polymerase recognizes the promoter region, the sigma factor is released. So you can see in this picture, this is the promoter region. Now, there is a bubble in the structural gene area. In the 5' to 3' direction, the mRNA is started synthesizing. Now only the core enzyme is present, which is required for the polymerization reaction. Sigma factor, after initiation, it left. As RNA polymerase recognizes the promoter region, the sigma factor is released. RNA is synthesized from 5' to 3' end by adding ribonucleoside triphosphates, GTP, CTP, UTP and ATP. The sequence of nucleotide bases in the mRNA is complementary to the template DNA strand. This already we know, right? The nucleotide sequences which is present on the mRNA is complementary to that of the template DNA strand, but it is same as that of the coding strand, where in place of T, U will be present in mRNA. No primer is required for RNA polymerase. When you look at DNA replication, RNA primer was required because DNA polymerase will not start its process unless and until an RNA primer is synthesized. Moving on to the next session, we are going to study about termination. This is the end process of transcription. Termination is of two types, row-dependent termination and row-independent termination. So first we will study about row-dependent termination. There is a protein factor. There is a specific protein that we will call it as the row factor. It will come and bind to the growing RNA or weakly to DNA. In its bound state, it will act as ATPase and it will terminate transcription and it will release RNA. Okay, so here you can see this is a newly synthesized RNA. This is the DNA. Now the Rho factor will come and bind either to the growing RNA or weakly to the DNA. Once it joins here in the bound state, this is acting as a ATPase, and what happens? The RNA is released. Okay. Now this step where we are utilizing Rho factor, so it is called as the Rho dependent termination. Next we are going to study about the Rho independent termination. When you look at Rho independent termination, it is because of the palindrome which is present in the DNA. Can you see here in the DNA? Here what happens? This is a GC rich area. Once the palindromes are formed on the mRNA, the palindromes will help in the formation of a hair loop. Once this hair loop is formed, further the RNA polymerase cannot move, so the RNA will be released. Okay. Rho independent termination. The presence of palindromes in the base sequence of DNA in terminator region creates hairpins in the newly synthesized RNA. Okay. So we studied about termination. There are two types, Rho dependent termination and Rho independent termination. In today's biology tips class, we studied about transcription. What do you mean by transcription? The process of formation of RNA or the process of copying of the information present on DNA to RNA is called as transcription. And transcription happens in a region of DNA called as transcription unit, which possesses three areas: promoter, structural gene, and terminator. Promoter is a region where who will come and bind? DNA-dependent RNA polymerase will come and bind. Structural gene is a region which carry information. Terminator is a region which helps in ending transcription. Thank you.